I would be talking about two things in this talk. Uh, First part of it, I'll talk about the work we did, me, Shakti, and Professor Tara did a few years back on the hierarchical networks, and then I'll shift gears and talk about the work we did on the human connectome. And there are some like overlaps and some relations. So I'll begin. So by now, I don't have to sell you why networks is important or network neuroscience is important. I guess by now you must have got ample and good amount of talks from Professor Labra and Shakti on why do we study networks. But I'll still quickly go through the reasoning that the brain is the most complex object in the universe. It's a complex system, and it functions via the dynamical interactions between its components. Now, these components don't interact with each other all at once at the same time. There's a structure to it. In fact, the interactions are highly structured, and those interactions are driven as well as constrained by their anatomy, what we call as a structural connectivity. So in neuroscience, a lot of focus is on the functional connectivity and the effective community connectivity, but not enough focus is on structural connectivity. And when I was working there, we have more focused on the structural connectivity because there's a lot to unpack there. So there's a lot we can do with the network neuroscience tools on these structural networks. And for example, the core periphery organization and the modular organization. These are the examples of large scale ROS organizing principles in a network, which Shakti has introduced in one of his talks. We were focused on one particular such organizing principle called hierarchy. Now, hierarchy is very misused and abused term in the neuroscience literature. If you look at it, there are like hundreds of papers about hierarchy, and to everyone, it means something else. Depends on whom you ask. So, I mean, there is a modular hierarchy, which means there's a nested modules within modules within modules. That's a hierarchy. Some people call core periphery network, core periphery organization itself as a hierarchy. And there are lots of hierarchical hierarchy defined based on the anatomy, where a lot of biological uh, studies, like biological information, is required. But we still don't. We until recently we did not have a specific paradigm to describe hierarchy. So first of all, what do we mean by hierarchy? So we had a very specific definition in mind. We meant hierarchy is an organization of networks into layers, uh, and those layers are in a sequence. And those layers, the consecutive layers, talk to each other much more frequently. So there's a dense interconnectivity between the consecutive layers and sparse connectivity everywhere else. So you can see a kind of a schematic there. And we are not dreaming that structure out of thin air. These kind of structures do exist. In fact, one of the oldest studied structures in brain is a hierarchical structure, even before I'd say modularity, I guess, because uh, if you look at the work of Feleman and Vanison, who studied the visual stream, and they delineated what are the specific levels of processing. There's a primary visual area, there's V1, there's V2, there's V4, and then there's an entire stream. And there's a directionality to it. So information travels, and there's an increasing complexity of processing. So that's one type of hierarchy. Another type of hierarchy we see in the cortical columns, where at a completely different scale, at the scale of neurons. The first one I talked about was at the scale of brain regions, where one brain region connected to another brain region through axonal tracts. So in the different scale of neuronal scale, you see the cortical columns, where the neocortex is very nicely arranged into different layers. All these layers are anatomically defined. They have different types of neurons, different types of connectivity, and those layers also have very like canonical structure of connection. What layer talks to what layer talks to what, and how the information goes from one layer to another. So these kind of structures do exist in the brain. We know through anatomy. The question is this something more universal? Is it ubiquitous? Can we find it at different scales in different species? If it exists, how do we find it? Where it's not obvious, where it's not anatomically very clear. So if you get something like this, how to get something like this? Now, this question is answered for modularity. For modules, if you give a network, there are n number of ways through which you can find clusters of dense interconnectivity, and you can find modules. But for this specific structure, we don't have any example. So then we had to first define what hierarchy means, and then a quantifiable quantity called hierarchy index, which is actually quite simple. It What it does is it assumes first a hierarchical structure. It sorts a network into the layers, and it sums up this quantity over the interlayer connections. So these the off diagonal blocks. This is the connectivity matrix AIJ, the agency matrix, and these are the in degree and the out degree, and L is the total number of connections. You might remember this. It looks very similar to the modularity measure. It's a twist to that. And once you define that, this is the quantity which we have to maximize. So we have to find a particular configuration which will give us the maximum hierarchy index. And the maximization is the real key here. How do you maximize it? So we, uh, I mean, it is a NP hard problem to find the absolute maxima, the global maxima of which exact. So we have to optimize not just for the sequence of the layers, but also the membership of the layers, what belongs to what. So first we have to group it and then arrange it in a sequence. And both of them should perfectly match so that we get the maximum hierarchy index. And if there are n nodes, there's a factorial n possible ways. I'm not factorial n, but yeah, there's a you can calculate how many number of ways you can uh, rearrange it. It's a very large number. So then we find approximations, and one of them is using simulated annealing, where we navigate the energy landscape. So H is the energy here, and we are navigating the energy landscape by changing the configuration minutely at each step, iteratively. And at each step, we either accept that change or we reject that change. That's the standard simulated annealing. And the schematic of that process is given here. So starting from a network which does not have any structure, we arbitrarily divide it into layers. And then at each step, we make one of these four changes. We either split a layer into two, or merged two layers, or interchange any two layers, their positions, or start a new layer by randomly picking one of the nodes and putting it out. So these four steps randomly we choose, and we navigate the energy landscape. And after large number of trials, we do actually get what we want. To test this on synthetic networks, we design these hierarchical networks where there's a dense connection between the layers. This is the agency matrix. And what the algorithm gets is this. It does not have any knowledge of the layers, but using that procedure, it manages to retrieve those layers like this. And you can do that to different degrees of hierarchy. So this one is more hierarchical. You can see it because it's much more sparse connections here. And that's why it's much more accurate. So this is the accuracy. How close is the obtained layers to the original layers? If you make it more blurry, it's more dense. You still get the hierarchy, but not as accurate, but it still gets something. So this kind of sets up the that the method works. And then we start analyzing the real networks, the macaque brain, the human brain, and the C. elegans. So this is what we see in the macaque brain. So if you run macaque brain through this pipeline, you get this thing, 16 layers. And there's a specific sequence and there's a specific membership of those layers. What you see here is the measure of how robust these layers are. It's a consensus figure. So each run of simulated annealing will not give you the exact same answer. They might vary because there can be degeneracy. And at each iteration might, in principle, give you a completely different membership of layers and completely different sequence. But that does not happen. What you see here is this is for the sequence and this is for the membership. And in both the measures, it's quite robust, which means uh, a collection of nodes say in the same show up in the same layer in most number of iterations, and those layers are also arranged in the same sequence. So it's quite a robust sequence of 16 layers. So far, so good. But we know already that the macaque brain is modular. In fact, Shakti has given a talk on that. So when we studied the modularity in the brain, we found there are 5 modules, and those modules are also very robust, very dense modularity and all. How do both these structures interact with each other? What's the relationship between the modularity and the hierarchy? So what you see here is uh, the alluvial diagram, which shows the comparison between the modular breakdown and the hierarchical breakdown, the layers. And it's a very good correspondence. What actually is happening is that each module is splitting into a few layers. So the 5 modules is showing up as 16 layers. So, which means that we were looking for hierarchy, pure hierarchy. We were not even looking for modularity, but we end up getting the modules also. So this modularity and hierarchy are deeply intertwined. And that's why we call it modular hierarchy. So there are already a term called hierarchical modularity. We are calling it modular hierarchy. So it specifically means a structure where there are modules and there are hierarchical layers, and they mostly correspond. There's a correspondence between modules and the layers. The question is, which uh, structure is stronger? Is the hierarchical structure is stronger or modular? What is the dominant structure? There could be 2 scenarios. One scenario could be that the modular structure is stronger and the modules are almost independent from each other. And within the module, there is a finer inner structure, which is hierarchical, which is so. What you see here are synthetic network, which is very similar to macaque network. They are built like macaque network to test these 2 hypotheses. One, where the modules are independent and they have a inner structure. In the other one, there are modules, but the hierarchical structure is more dominant. By that, I mean is that between 2 modules, 2 specific layers are kind of tethered to each other. So it's like a chain. So if there's a global hierarchy, which is the strong influence here, it should be something like this. And if it's modular hierarchy, as we call it, it should be something like this. And then when we run the algorithm in this, what we see is that the modular membership is kind of robust in both the cases, but the sequence, and it's very counterintuitive. One would imagine that this scenario should give a very robust sequence. But what you see is that this is the one which gets a more robust sequence, even though the modules seemingly were independent. And in this case, this is confusing the hell out of the algorithm. It's the modules and this part is confusing it completely, and it's scrambled all over the place. So clearly, this is the scenario which is closer to reality. So this modular hierarchy actually opens up a lot of questions about this kind of structure. Why this kind of structure? What does it do? So both have theories, and until now it's just theories and speculation. We don't know yet. But the way we are interpreting it is that different types of processing models about brain. So there is distributed processing, and then there is a integration. They don't need to like, necessarily they don't contradict each other, but they both are focused differently. So the modular structure seemingly supports the distributed processing, where in different components of the network, different information processing is happening parallelly. Whereas in this integration picture, things happen linearly in a sequence. So possibly brain is tuned to do both. It does both. Uh, and there are examples of such architecture already. For example, the cortical columns in visual cortex does just that. There are functional modules in the cortex which are independently processing different parts of the scene which you are seeing, but then within them there is a hierarchy which is integrating stuff. But the fact that those modules, even though they seem independent, there is a global sequence. It's not apparent from here, but there is a global sequence that shows that both these processes are somehow nicely integrated. I mean, evolution has developed it in a way that both the things are happening in tandem. Another thing is deep versus shallow architecture. So there's a body of literature which talks about how the artificial neural networks are inspired from brain, and there can be lots of correspondences of how they process uh, visual pictures and whatnot. So there also uh, there's this deep architect. How deep is that those layers? Is it just 3-4 layers, or is a deeper one? And it seems like it has both. It has shallow structures as well as deep structures. So, and one can go deeper and find more correspondences, and can work more towards this. I mean, this is just an opening of a new door itself. So once we know that this structure exists, where all it exists, like is it just a macaque, or it exists ubiquitously in all many species? So we did that with the human network. So this is a human connectome. Uh, we used a specific data set, which I'll come to later. It has some 188 regions, and it has the brain connections are discovered using diffusion tensor imaging. It's like fMRI, but for static images. So this also has a very robust hierarchical structure, modular hierarchy. And then we switch scales and go to C elegans, which is just neurons, 300 neurons. And even that has uh, the modular hierarchy. So not just it's conserved across species, but it's also conserved across scales. It's a very, very ubiquitous structure, and it should be studied more. So an interesting thing came up while looking at the C. elegans, which is if you look at the breakdown of the layers, it's a sequence. And if you look at the breakdown of layers according to cell types, so sensory neurons, interneurons, and motor neurons. There are three types of cells in C. elegans. Sensory neurons gather the sensory information, the light, the chemicals, the mechanical stimulus. And the motor neurons are the actuators, like which allow the animal to move around and change its shape and everything. So you can see that the sequence is, uh, I mean, the sensory neurons. So the sequence of neurons is very much in accordance with what you'll expect. That there should be sensory neurons, then the interneurons, then motor neurons. So a good structure function correspondence here. So that's the big picture of this work. Uh, it's a fundamental form of organization which was not adequately addressed until now, and we addressed that issue and developed a method which can uh, quantifiably be find the hierarchical levels. And by doing that, we ended up finding a more complex entangled organization, which we call modular hierarchy. And that modular hierarchy contains very intriguing possibilities of implications of what kind of role it might play. And yeah, and it also kind of builds a bridge between the artificial neural networks and the brain. So for those who are interested, the paper is out and you can look it up. Uh, so that was about the hierarchy. And now I'll talk about the other work which we did, which is kind of, I don't know, it we found some very interesting statistical patterns in the brain connectivity data. And it's one of those things where we find something very interesting, but we don't exactly know what it means. But it is something which needs to be told. So the human structural connectome uh, is obtained using the diffusion tensor imaging, and this is how it basically looks like. So you are finding out the white matter. Uh, the brain regions are connected to each other through axonal tracts. They are like thick cables. And one of the challenges while studying this is how do you parcellate the brain? Because there are n number of parcellations. And some atlases have 700 regions of interest, some have only 50. So we chose a data set which has this parcellation, Craddock et al., which has 188 brain regions, and it is based on uh, the resting state network from MRI, fMRI. The reason we chose this was it had a very large human population. The number of subjects was quite huge, 196 healthy subjects. So we can ask questions about a healthy brain, like how a brain exists, like what is a typical brain like? That's the question. So it's an undirected network, unlike the macaque, and but it's a weighted network. So they have assigned a weight to a connection based on how thick the axons look like, and that's all. There's nothing else. How thick the axons are. So it's a weighted network from a large population, and that allows us to ask all sorts of questions. So this is just we lay down the data how it looks like. So these are the connectivity matrix, and how are the weights distributed? So this is how it looks like. There are two metric we assigned the weights. One is how frequently a connection occurs. Does it occur in all the individuals or does it occur in only few? So there's a fraction of the frequency of occurrence. And then what's the connection weight of those links? So you can see that from 0 to 1, 1 being it's a universal connection. And you can see that as it approaches 1, it has a completely different distribution. It's like a completely different set in itself. And these are the links which are not universal. But we observe that there is a strange connection between the frequency of occurrence and the average weight. So when we average it, the average is only across the population where it exists. So we are excluding the zeros. Zero means it doesn't exist. So this relation is quite non-trivial, that the existence of a connection and what's the weight of the connection should not have an obvious correlation. They both are different process. The process through which the brain is wired during development and the process through which the strength of the connection is determined. They are different processes. We found that it follows a very interesting statistic, and that allows us to combine both these the frequency of occurrence as well as the connection weight, which is that a given connection across the population follows a Poisson statistic, Poisson distribution. And this is the Poisson distribution. It's a very specific kind of distribution meant for independent events. It's so these are integer. The random variables are integers. The connection weights itself are not Poisson distributed. They are actually a product of a Poisson random variable and a scaling factor. So this is the weight which the W is the weight which is measured in the experiment, and this is the scaling factor, and this is the hidden the Poisson random variable. And from this, you can find both these quantities. You can find the scaling factor and you can find the weight. And what we find is that majority of the connections follow the Poisson statistics. Each of them have their own lambda. Lambda is the average as well as the variance. So this is the example of 4 different connections. So some of them have a low lambda, which means they'll have lots of zeros. Some of them have high lambda. They won't have any zero. So it accounts for existence and non-existence of connections also, and the weight distribution also. One thing to note here is that there is no correlation between the Poisson parameter and the scaling factor. They both are different things. And they both might have different biological interpretations. What do they mean? So our hypothesis is that the Poisson part, the integer part, might actually represent the connection strength. How many exons are there in a particular cable, in a particular tract? That decides how strong is the connection. And the scaling factor might be some peripheral factors like what is the thickness of the myelin sheath, not the number of exons, but each exon, how thick is the coating, and that also adds to the overall thickness of the cable. So if once you make that hypothesis that this means something, the rescaled Poisson variable means something, and not the measured weight, how do we test it? Uh, the only way we have is to measure it against the functionality, the functional connectivity. So luckily, this data set also had the functional connectivity, which means that it's a dynamical measurement. So if two areas are functionally connected, means their activity is very similar over time. So that means they are somehow connected, either directly or indirectly. So this is the set of functional. So for each individual, you have the functional connectivity, and you can say that it's much more denser than the structural connectivity. And we know that the functional and structural connectivity don't have a straightforward relation. I mean, we don't know. It's a hard problem. But you can still calculate some simple quantities like correlation. And the correlation will not be high, but it'll have some correlation, some significant correlation. So on the X and Y axis, you see the comparison of structure function correlation, where in x-axis the structural uh, connectome is what is in the data, the measured weights, whereas in the y-axis it is the Poisson variable which you found out. And what you see is for every individual, not some, not most, every individual, the correlation is improved when you use the rescaled weight matrix, not the original weight matrix. And that's a very unlikely thing. I mean, which means it's a significant effect. Of course, I mean, you can't have a very high correlation just by doing rescaling, but at least you're going towards. I mean, you're going towards the functionality. So it is more biologically meaningful quantity than the actual weight. Now we go even further. Uh, Before previously we only compared the structural weight matrix and the functional weight matrix. Now we try to simulate the functional weight matrix from the structural connectivity and then compare it with the actual functional connectivity. So how do we simulate it? We use a neural mass model. We take the Wilson-Cohen model. It's quite popular. And each node is a Wilson-Cohen oscillator, and you connect them using the same weights as the structural weight matrix. You get some dynamics over some time, and then you find the functional connectivity. It's simply correlation between the two nodes. So these two are the simulated functional connectivity from two different structural weights. One is the observed weight and one is the rescaled weight. And this is the empirical functional connectome. When you do this comparison, you again see the same thing. That using the rescaled structural weights, you see an improvement in the correlation. And for a single individual, the improvement does not look like much. But when you look like at the whole population, and you see that the entire population has improved, that's a very significant effect. So both these things very strongly suggest that the rescaled Poisson variable, which is hidden inside the weight, which we found out only using the population statistic, is somehow giving us something biological. And there's another interesting fact about this is that the entire network is actually two subnetworks, and both these subnetworks has completely different statistics. So, and it's a if you look at the variance, the coefficient of variance of each link, it's highly bimodal. So what we did here was we divided this network into two. So remember here we saw that the links which exists everywhere, they have a very different distribution. So that's what we made as a criteria. The ubiquitous links which have which are occurring 100% of the population. That's one set, and rest all the links are a different set. You can see that the variance is completely different. And then lambda, which is directly related to the coefficient of variation, they also have a bimodality. So there's one network which has very high lambda, and there's one network which has a very low lambda. And we call this as a basal network, which is like a backbone of the brain. It's something very essential without any variation so that the brain can function. And the rest of the links are kind of peripheral, and we call them superstructure links. Now, that's a conclusion we are reaching purely from math. I mean, and that's not enough. We need to do more biology on it to say something more. But clearly, there is some pattern here which needs to be looked into. Note that when you do this with the the rescale factor, which I said is corresponding to the thickness of the exons, that does not have any bimodality. It's same for all the networks. So that something is not affected by whatever is happening here. So in conclusion, there are 3 important things from this study. One is that it follows a very non-trivial statistic, Poisson statistic, which allows us to explain the frequency of occurrence of a link also, and the distribution of weights, which means it implies something about the generative mechanism. During development, how is the brain formed? And other thing is using a population analysis, we are actually able to filter out an important quantity from the observed weights, which has some biological interpretation. And then there are 2 subnetworks in the brain, which wasn't obvious, and they're clearly different from each other. Why are they different? That's something to be studied. But there's a clear difference between the connections which exist in all individuals and the one which has a variance. So this was quite a unique kind of study which we did on a population. We didn't use any traditional methods available, and that's why we think it's a very important piece of work and it needs to go out. So yeah, these were the 2 uh, things which I want to talk about, and I hope I managed to convey it. So thank you for your attention, and rest of the time I'm happy to answer any questions.
Thanks a lot, Anand. Actually, so we have lots of time for questions. I noticed that nobody asked any questions during the talk, so please do so now. Go ahead. Yeah.
Uh, that was quite an interesting talk. Like, I'm very curious to know, like, from like you worked on the human brain connectome data. So from the from the place where you get the data, you directly got the connectivity matrix, or you got those fMRI and DTI files, then you process it to find the connectome.
No, so both the data are required. Both are experimental data. So both are required. Both were there. There was a connectivity matrix, the structural connectivity and the functional connectivity. Both are measured differently.
And then you process it to find the connectivity matrix.
So we processed the structural connectivity matrix. So we had a structural connectivity already. It had weights and it had the topology. And using this rescaling, we processed it and found a better structural connectivity matrix, which had a better correlation with the functional connectivity matrix. Functional connectivity was given.
Okay.
Then we simulated a functional connectivity from the structural connectivity. Like if suppose your brain was Wilson-Cohen oscillators, which it is not, but we need some dynamics. So comparing a fake dynamics with the real dynamics and how close they are. Does it answer it?
Yeah, thank you.
Okay.
The slide that you mentioned Poisson statistics and the scaling factor. How did you do that breakdown? How do you know it's a,
How did you?
like how do you know it's a cumulative effect of these 2 processes?
Oh, okay. So okay, I kind of I didn't show all the figures. So if you look at the archive, the hint came from when we plotted the variance and the mean. They were nicely falling on a straight line and with a slope 1. So that is a signature of a Poisson variable. But that quantity itself was not following a Poisson statistics. So you have to multiply the entire distribution with some constant to bring it to a Poisson distribution. So that's why we know that we hypothesize that it's a product of a constant and a Poisson variable. So yeah. So if this is the case, then you can find this and this. So we found this, and then we see that this thing nicely fits Poisson variable. So we use chi-square goodness of fit. So for each link, for each ij, most of them, you get this. Some lambda you get, for which it fits very nicely. And the details of how we like did the goodness of fit and how we found lambda and all is in the paper.